This is the Nutrients lecture with a focus on phosphorus for ENR4345, Methods in Aquatic Ecology course at The Ohio State University. In this lecture, I will go over the importance of phosphorus in aquatic ecosystems, as well as different types of phosphorus that can be measured. Colorimetry, which is a method for analyzing freshwater samples for phosphorus. Quality assurance and quality control of phosphorus samples, and finally, data interpretation. Phosphorus is an element that is required for all life on Earth. Phosphates are the bioavailable form of phosphorus, and generally, phosphorus is a limited nutrient in a healthy aquatic ecosystem. A natural source of phosphorus is the weathering of rocks, which is a very slow process. In modern times, there has been an anthropogenically created excess of phosphorus in aquatic ecosystems, which can lead to negative environmental effects, including, but not limited to, harmful algal blooms and dead zones. Excess phosphorus can enter an aquatic system as runoff from farms, industrial waste, or sewers and sewer overflow. In addition, anthropogenic activities can increase internal loading, which means that the phosphorus that was stored in sediments is being released into the water column where it becomes bioavailable. I want to mention that the best solution to this problem is not to place blame on farmers, sewer operators, or others. We all benefit from these goods and services, so the best response is to work together to create a solution that works for all stakeholders. For example, when helping a farmer, creating a solution that both reduces phosphorus runoff and saves the farmer money on fertilizer is more likely to have a long-term impact than pointing fingers and placing blame. As an example, we are going to look at the Lake 226 experiment that took place in Canada and was published in the journal Science in 1974. In this experiment, scientists placed an impermeable barrier down the middle of Lake 226 to create two separate experimental areas that were experiencing the same environmental conditions. The same amount of nitrogen and sugar was added to each side of the lake. However, one half of the lake also received phosphorus. The side of the lake where the phosphorus was added experienced a harmful algal bloom, but the side without the addition of phosphorus did not have a bloom. This experiment helped scientists understand the link between phosphates and harmful algal blooms, and lead to the implementation of the Clean Water Act, including the ban of phosphates in household detergents. The method that we will be using to analyze phosphorus samples in this lab is called colorimetry. Colorimetry is a standard method where reagents are added to water samples containing an analyte. The chemical being studied will react with the reagents to create a color. This method is used for a variety of nutrients, and generally, different reagents are needed for each analyte. More specifically, we will be using the molybdate blue method for analysis of soluble reactive phosphorus. The phosphorus within the sample will turn blue when it reacts with the reagents. The blue color will be proportional to the concentration of phosphorus within the sample, so a more blue sample will contain more phosphorus. After the reaction has occurred, we will be reading phosphorus samples on the spectrophotometer at 880 nanometers. The reading that the spectrophotometer measures is the absorbance, which by itself isn't very informative. We will create a standard curve containing standards with known concentrations of phosphorus, as well as the absorbances that are read for each standard. We will compare the absorbances of our samples to the standard curve in order to determine their phosphorus concentrations. As an example, let's say that I made this standard curve, and then I read the absorbance of one phosphorus sample to be 0.25. I can compare my sample to the standards containing known phosphorus concentrations to find the phosphorus concentration of my sample. I can use the equation of the trend line on my standard curve to mathematically calculate the phosphorus concentration of my sample. The R-squared value tells me how good a fit the regression line is to my data set. Most professional laboratories are looking for an R-squared value containing at least 3 nines, or 0.999 something else, to show that the standard curve being used is accurate. Notice that the absorbance, which is the value that we measured on the spectrophotometer, is on the y-axis, and the phosphorus concentration, which is the value that we are solving for, is on the x-axis. The next lecture in this module will have more information on creating and using a standard curve. If you go on to work in a water quality laboratory, it is likely that you will have a computerized autoanalyzer to process phosphorus and other nutrients from water samples. On this slide, there are 3 examples of different autoanalyzers that are commonly used. These machines are able to process a lot more samples in a shorter amount of time than could be done by hand, and they also help to reduce human error within the samples. However, it is important that the technician who operates these machines understands the chemical processes for analyzing nutrients, which is why we're doing this analysis by hand in class. The technician needs to be able to troubleshoot any issues with the machine, as well as make reagents and standards. The technician needs to be able to interpret the results by understanding a standard curve and perform quality control measurements on the samples run by these machines. Different sample preparation techniques allow us to look at different fractions of phosphorus within our sample. This bottle represents water collected from a grab sample. We will call this whole water because it contains all of the different fractions of phosphorus that were in the water at the time of collection. The different parts of the sample are important to know because they vary in their bioavailability. For example, a phosphorus molecule that is dissolved in the water would be easier for algae to use than one that is chemically bound to a piece of rock. The first fraction is total phosphorus. This whole water can be digested. The digestion process will turn all phosphorus in the sample into reactive phosphorus, which can then be measured with the molybdate blue method. Next, we can filter the whole water sample using a filter with a pore size of 0.45 micrometers. There are a few different ways that we filter water for this course, including a syringe or using a vacuum pump with a filter flask. The filtered water can be analyzed with the molybdate blue method, which will allow us to study dissolved reactive phosphorus. Or the filtered water can be digested and then analyzed, which will give us total dissolved phosphorus. Finally, there are forms of phosphorus that we can calculate after analysis is complete. The first is particulate phosphorus, which is total phosphorus minus total dissolved phosphorus. The second is dissolved non-reactive phosphorus, which is total dissolved phosphorus minus dissolved reactive phosphorus. QA/QC is a common abbreviation for quality assurance and quality control, which incorporates several practices that ensure that your results are accurate. QA/QC practices can help to discover and eliminate sources of contamination or interference with the chemistry. There are a lot of different ways to do this, and not all practices are appropriate for all analyses. When starting a new study, it is important to consider what QA/QC would be good to include in the study to help validate your results. I'm going to talk about 3 different QA/QC metrics that you may want to add into your studies. The Ohio EPA has regulations for which and how often these samples should be performed. Generally, it is suggested that QA/QC is performed at a rate of 5% of the total number of samples. I also want to mention that although this lecture is focused on phosphorus, there are QA/QC samples appropriate for all different analyses, including for analyzing other chemicals in water and for the ash-free dry mass and chlorophyll A analyses that we did in a previous lab. The first quality control is a blank. A blank sample contains DI water and should read as nothing, or in this case, no phosphorus. When a blank sample is not zero, it indicates that there is some kind of contamination that could skew the results of your samples. A field blank is when the blank sample is collected in the same way using the same equipment that all other samples are being collected with in the field. And a laboratory blank is DI water analyzed the same way and with the same machine as the water samples. Next is a duplicate sample. This is when two samples are collected or analyzed at the same time and place. These samples should have the same results or within 10%, as there may be some small differences in the sample that the machine can pick up on. If they don't have the same results, this could indicate an issue. A field duplicate is when two samples are collected at the same time and with the same equipment. A bottle duplicate is when a sample is analyzed twice in a row from the same bottle. A matrix spike is when a water sample is spiked with a known concentration of the chemical we are looking to analyze. So for phosphorus, you could add 1 ml of your high standard to 9 ml of your sample A and run this as a matrix spike. You will compare this spiked sample to the results of sample A that was not spiked, and the difference should be exactly what you spiked into the sample. This can help look for interference in the samples. Our standards are made using only phosphorus and deionized water, but sample A was water collected from a natural environment containing not only phosphorus, but several other chemicals and minerals that may interfere or react with the analyte or reagents in unexpected ways. Think about the interpretation of your phosphorus samples. The form of phosphorus is important, as well as its concentration within your sample. For example, you can have a sample that has a high total phosphorus, but low dissolved reactive phosphorus, which is a bioavailable fraction of total phosphorus. Next, we want to consider what is growing in the water we are studying. Lake Erie, which is notorious for harmful algal blooms, generally has a very low dissolved reactive phosphorus levels. That's because phosphorus is a limiting nutrient in Lake Erie, and therefore, algae takes it up as soon as it becomes available. This low phosphorus could be incorrectly interpreted as an indicator of good water quality, when in reality, all the phosphorus is located within the algae rather than dissolved in the water column. Finally, it is important to consider the timing and number of samples that are collected. This can vary depending on your research question. For example, one single grab sample could be very important if we are looking for something like whether or not the water contains E. coli. But one sample is not going to be very useful if you are trying to understand stream dynamics. Perhaps combining phosphorus analysis with another technique that we learned in class this semester, such as ash-free dry mass, or a collection of grazer macroinvertebrates with a DNet, could be a creative way to answer your research question.